the cardiac project is basically an international retrospective study that spanned across 6 tertiary centers, 3 different countries, looking at the cardiac complication landscape following the CD19 CAR T therapy for large B-cell lymphoma. The published report thus far indicate that the cardiac complication that happened in the context of CD19 CAR T therapy range between 5 to 15%. However, most of the published report do not address the particular complication, cardiac complication per CAR T products, and it does not compare the CAR T products among each other with regards to the cardiac safety post administration of these products. And what the primary goal of this study is to first provide a large descriptive analysis of the cardiac complication landscape post-CAR, and then compare different CAR T product and establish the differences between them, and if there is any distinct signal with regards to the safety and toxicity, and eventually reaching to establishing a differences in product selection for patients, particularly patients with significant cardiovascular disease or at higher risk of having cardiovascular disease post-CAR T cells. So the study included more than 900 patients distributed among the 3 FDA-approved CD19 CAR T product for lymphoma. Most patients received axi-cel, and the second most common was liso-cel, and then followed by T-cell. And the incidence of cardiovascular comorbidities or cardiovascular risk factor in the included patient was very high. So about at least half of the included patient have 1 or 2 cardiovascular risk factor coming into CD19 CAR T therapy, and about 1 in 5 patients, or almost 20%, have more than 3 or more cardiovascular risk factors, indicative of real-world nature of this study and indicative of the patient that we see every day, who are usually slightly older and have usually 1 or 2 cardiovascular risk factor. The incidence of cardiovascular complication between the start of lymphodepletion on day -5 and day 30 post-CAR T treatment was 9% in our study. We observed a slight increase in the cardiovascular complications in the T-cell compared to other products, at more than 25% in the T-cell compared to other products. However, we also observed differences in the type of CV events that happen after each product. The most common cardiovascular complication was arterial arrhythmias, manifested by atrial flutters and atrial fibrillation. Supraventricular tachycardia was the second most common. And interestingly enough, we also saw differences in the median time to the first cardiovascular events among different CAR T products. For example, while the T-cell was the earliest median time to the first cardiovascular events was 5 days post infusion, was the latest median time to first cardiovascular events in the liso-cel cohorts, up to 9 days post infusion. We also observed that the axi-cel and the T-cell cohorts cardiovascular event cluster early post infusion in the first week, and follows an early peak and then kind of slow down after that, while the liso-cel follows a more peakless flat pattern of cardiovascular complication that goes all the way up to the 1 month after the CAR T infusion. And finally, we since this is a large group of patients who are heterogeneous in their characteristics, we performed a propensity score matching analysis in which we matched patients together and to decrease the heterogeneity of this cohorts. And we adjusted we identified multiple covariants that we adjusted for in the propensity score matching analysis that include age, functional status, tumor burden, and the number of pre-CAR T cardiovascular risk factors that we talked about in the beginning. And then we compared the cardiovascular safety of each CAR T product using the propensity score matched analysis. The first one was comparing axi-cel to T-cell. T-cell was associated with a higher cardiovascular complication within the first 30 days post-CAR T infusion compared to the axi-cel in an adjusted logistic regression model and also in propensity score matching analysis. And the second comparison was between axi-cel and liso-cel. And in that part, liso-cel was associated with lower cardiovascular complication post-CAR T infusion within the first 30 days in an adjusted logistic regression model or also in propensity score matching analysis. And the last thing, we looked at the overall survival of patients who developed cardiovascular complication in the first 30 days post-CAR T cells to those who did not develop cardiovascular complication. And the overall survival was lower in the group who developed cardiovascular events, highlighting the significance of this complication and the impact overall on the outcome, with about 10% difference overall survival by the end of first year. And this overall survival signal was mostly driven by an increase in non-relapse mortality. Despite that was not statistically significant in our cohorts, the patient who developed cardiac events within the first 30 days post-CAR T cells was demonstrated an increase in non-relapse mortality by the end of first year. I think the conclusion of this study is that many of our patients that we treat have a significant cardiovascular comorbidities, and particular attention should be paid to optimizing the cardiac safety on these patients, this group of patients, and involving and engaging the cardiovascular expert in that regard is important to mitigate the toxicity. And then our data suggests that results suggest that it may actually inform product selection in a sense of patients with pre-existing cardiovascular disease or patients with a significant higher risk of developing cardiovascular post-CAR T cells. Liso-cel appears to be safer compared to axi-cel.